You know, the first I thought would be if you could review your fantastic Nobel Prize winning work, which, you know, the more I read, the more fundamental it becomes. And I realized, correct me if I'm wrong, you know, I realized that ribosomes use up a large part of the energy of every cell in the body. So
yes, well, construction always requires a lot of energy, you know, so much more than destruction, I would say.
So the more I read and learn, you know, the more fundamental your work becomes.
Yeah.
So what I thought is maybe in your own words, you could explain a little an overview about, you know, you have so many things, the structure and, you know, the function and the speed.
Sure. So, you know, one way of looking at life is propagation of information.
Yes.
You know, so we have information in our genes that tell a cell how to grow, how to do all its activities, but also how to grow into an organism, you know. So all of that is coded in our genes. There's a program.
Yes.
But instructions by themselves won't do anything. You have to somehow be able to use those instructions to do the things that the instructions want you to. So it's a bit like if you have either a recipe or a blueprint, okay? By itself, it doesn't do anything. Something has to use the recipe to make the product, right?
Yes.
And so in all our cells, there's a very ancient, large molecular complex called the ribosome. And it's has 500,000 atoms, so it's huge in molecular terms. And what it does is, when you need to make a protein, the instructions to make the protein reside in our genes. So for every protein, there's a gene that specifies how to make it. And that portion of the DNA that contains that gene is copied to a sort of temporary molecule called messenger RNA.
Oh, messenger.
Yeah, you know, you can think of it, you know, if you wanted to go to the British Library and look up a very rare book, the chances are they're not going to let you take the book, okay? But what they will do is give you a copy of the book. And then you can read the copy at your leisure, right? So it's the same thing. The cell contains its genes on DNA, but when it needs to use the genes, it makes a copy called messenger RNA. And it's that copy that's read by the ribosome to make proteins. And there are thousands of proteins in every one of our cells, and each cell type makes a different combination of proteins. So your skin cells don't have exactly the same proteins as your red blood cells or your liver cells, you know. So there's a lot of specialization on which proteins are needed in each cell. And what the ribosome does is make all of the proteins. So I like to joke that, you know, first of all, it's one of the most ancient molecules. It existed even before there were proteins, before life as we know it existed. So it comes from an ancient form of life. And the second thing is that virtually everything in our cells was either made by the ribosome or made by other molecules which the ribosome made.
Okay.
You know, so it's sort of the mother or grandmother of everything in the cell. Okay. And now all of this was known in the 50s and early 60s. But what people didn't know is how it did it. How did it actually read the code? How did it put together a protein? How did it move along the messenger RNA? How did it know where to start and where to stop? So all of those things required knowing what it looked like. You know, if you want to understand a large machine and you don't know what it looks like, you already have a huge handicap. So what the work that we and others did was elucidate the atomic structure of the ribosome, which then led to understanding how it carried out many of those steps.
You know, one thing which I was puzzled, which you don't talk much in your book or in the papers I found is how fast the ribosome can do that. And I read that there are some issues about the energy, but then there's also the entropy issue, and which is related to the movies both you and also
yes.
Science show at the end
the
of
movies are a bit of science fiction, I have to tell you. So if I were to show the real movie, it would look like a mess. Okay.
So you picked the most important parts. Yeah.
I picked it as if only the right tRNA molecules, the right amino acids or building blocks are brought into the ribosome and move on. But in fact, what happens is when the ribosome moves along the mRNA, there's a code on the mRNA.
Yes.
It tells the ribosome, now you should select this amino acid.
Yes, yes.
Okay, out of 20 different amino acids. Yes. And so how does a ribosome know how to do that? Well, it looks for a match between this little adapter molecule and the code on the mRNA. But that means the wrong ones are coming in, and the ribosome says, this is a mismatch, and I'm going to reject you. Okay? And if it's a correct one, it immediately accepts it, very quickly accepts it, and then moves along and makes moves on to the next piece of code and incorporates the next amino acid. So this all happens very fast, 20 amino acids per second in bacteria. And even in higher organisms, at least a few amino acids a second. And that's much faster than any protein synthesizer that we can make in the lab, a chemical synthesizer. And it's also far more accurate. It only makes about 1 in 10,000 residues as a mistake, okay? So you have to realize the ribosome is not only very fast, but also very accurate.
Yes.
Talk about the entropy problem. Well, that's because it's using a lot of energy to drive this reaction in a unidirectional way.
Yes. Oh, okay. And the energy, where does it take the energy from?
There are two places. One is that when the amino acids are added to the tRNA molecule that brings the amino acid to the ribosome, that's a high energy bond. So energy is consumed even to make that bond, okay? So when that amino acid is released, there's a release of energy, okay? So that's one source of energy. The other is that many of the factors that bind to the ribosome, even the factors that bring the amino acids to the ribosome, the factors that help the ribosome move along the mRNA, all hydrolyze a high energy molecule called GTP. And the energy released from that is somehow used by the ribosome to make it unidirectional.
The other point I came across, which others talk about, and I'm sure you worked about this also, but I couldn't find the papers where you wrote about it, is how the ribosomes are created. There are so many.
That's a huge problem. And I have to say, we have not worked on it much at all. We accidentally discovered, we trapped ribosomes that were partially assembled. And we figured out some things about a stage of assembly. But what you're saying is very interesting. I said the ribosome is 500,000 atoms, but this is actually, it's made up of several large pieces of RNA, but about 80 proteins in higher organisms. And even in the simpler ribosomes of bacteria, it consists of 50 proteins bound to these RNA. So in the 70s, a Japanese origin scientist, I should say, Masayasu Nomura was working in Wisconsin. And he showed that you could take all the proteins apart in bacteria and reassemble them in the test tube, just by mixing them together. So people thought, well, you didn't need anything special to assemble it, that this would be a self-assembly process. But actually it turns out that's a very inefficient process that doesn't generate a lot of really active, very active ribosomes. It does generate active ribosomes, but not as good as the cell.
Okay.
In the cell, it's different. In the cell, what happens is there are lots and lots of proteins. And what they do is they stop the assembly at different points to allow reorganization of the structure. And then they leave, and then it reaches the next stage, and there are other factors that bind. So it's a highly orchestrated process in the cell. So it's almost as if there's an entire factory to assemble what itself is a factory, okay?
Yes,
so you can think of the ribosome as a large factory to make proteins, but there's a whole factory to assemble the factory. Of course, that's like real life, you know? If you want to have a factory, you have to have something that builds the factory, right? And then the factory can build whatever it's supposed to build. So it's a bit like that. And the study of ribosome assembly is very complicated. And it's being studied in lots of labs. And one of the methods that's really helping the study of ribosome assembly is this new method of visualizing structures called cryo electron microscopy.
Yes, you mentioned it. Yes, yes.
And the reason for that is you can look at assembling ribosomes, and computationally you can sort out which ribosomes have reached which stage of assembly, separate them, and then figure out what the assembly process must look like. So a lot of work is going on. It's a very active area.
If it's cryo, do the ribosomes work at these low temperatures?
No, they're trapped. They're trapped. So they were working one instant, and a few milliseconds later, they've been plunged into liquid ethane. So they're trapped in whatever state they were in. Okay, so you can get a whole population of states from.
That's this like a cooling trap, which you describe in the book, where your
a little bit like that. Yeah.
Like 3 months.
That didn't work out so well.
So you say that the creation of the production of the ribosomes, is that solved now, or you say this is still a very active area?
No, the assembly and the biologists prefer a term called biogenesis, which is to say biological creation of the ribosome. That's a very active area still. Okay, it's people are figuring out a lot of things about the pathway, but as I said, it's a complicated process that involves maybe 100 different players that are involved in assembling the ribosome.
Yeah, I read that there are mechanisms also to dismantle incorrectly assembled ribosomes.
Exactly, and that's part of the assembly process, is that if you somehow manage to get an incorrectly assembled ribosome, the cell has to recognize it and get rid of it.
And how long do they live? In this one article.
People have asked me that. You know, I'm embarrassed to say I don't know. You know, there is ribosome turnover, but I don't really know what the lifetime of a ribosome is. And every time somebody asks me this, I resolve that I should go home and look it up, but I always forget. It's such a good question. Maybe this time I will go and look it up.
When I put this movie on YouTube, we'll probably get many applications for PhD students wanting to work on it with you.
No, actually, you know, it's interesting. The people who apply to me have a particular interest in mechanism and structure. Those are the sorts of people who apply to my lab. They're not sort of, you know, if they're sort of just generally vaguely interested in molecular biology, they probably won't. And if they want the next big trophy, they probably won't come to my lab.
So what you're working on now is crystallography and structure. And
we're mostly using electron microscopy now because it's almost superseded crystallography for very large structures. Of course, for medium and small structures, you still need crystallography. But the very large structures are so hard to crystallize that, you know, it seems as if, you know, you could get almost as much information about by electron microscopy. And sometimes, and actually more powerful You could do things with electron microscopy that you could never do with crystallography.
Okay.
For example, trap these very rare states and so
on. Okay. By cooling them at the right time.
By cooling them. And you can also classify, if you have a mixed population in your sample, you can computationally separate out all of the individual groups of structures.
Okay.
And solve, so you could get many different structures out of just one experiment. You know?
Okay. If you like, we can switch to the next topic we thought of discussing, which is is it your main work now, main responsibility is the Royal Society, as president of the Royal Society. I heard that...
Yeah, I wouldn't say it's my main responsibility. I mean, the Royal Society presidency is an honorary job. I'm still paid by the MRC to do ribosome research.
Oh, I see.
So that's my day job.
But it's responsible for all of British.
Exactly,
science.
But it is a serious responsibility, I would say. Yeah,
also,
but it's an honorary job. It's not a salaried position, if you know.
But you make headlines in guiding.
Yes, no. I have to, when I took the Royal Society job, I was actually, first of all, I was very surprised to be asked if I wanted to be considered for the presidency.
Well, I talked to some of my friends in Cambridge, and they think that it's a very, very good choice to have you, because
they're very kind to say so. But I'll tell you some reasons why I would not have been a good choice. And one is that I didn't grow up in Britain, and wasn't educated in Britain, so I didn't have the broad contacts and network as someone who is an establishment scientist in Britain would have. So I was definitely not part of the British establishment.
Okay.
And the second is that when I came to Britain relatively late in life, and I kind of parachuted into the MRC Laboratory of Molecular Biology, which is a bit of an island, even in Cambridge. You know, it's a very autonomous institute. It's not really part of the university, although it has links.
It started in the Cavendish, you know. I still have
but that was a long time ago. And even in the Cavendish, it was not funded by the physics department. It was funded by the MRC unit for molecular structures, you know. So it was housed in the Cavendish, is probably more accurate to say. So I would say that because of that, I was a bit surprised, and I didn't have any experience in policy or in sort of the broader aspects of science. But so I warned them about it, but they didn't seem to care. So I said, fine, I'll give it my best shot. Now, I talked to Paul Nurse before accepting it.
He was the president before you, I think, no?
Exactly. And I would say Paul said, look, everything looks good. The government is very well disposed towards science. You'll be a sort of cheerleader for science, and it'll be a very interesting and pleasant sort of job. And that was fine. But then 6 months into my presidency, the UK public decided to vote to leave the EU. Okay. And that has really serious consequences for science and how our long, extensive collaborations with Europe, how would they work out? And would we be perceived as insular? Would we continue to attract very good people to Britain? So these are all big questions. Suddenly I was thrust into the middle of what was a really quite political situation. And then, in the last many months, we've been hit with this pandemic. So suddenly I found myself, convening a group to look into various issues with the pandemic and to provide input, to government advice for the pandemic. So this is not something that I had ever planned on, but it just shows you that when you accept a position like that, you can never know what's going to come your way. You simply have to, play it as it comes.
But to me, it also shows the strength of the British science system, that they pick the best for the position, globally they can find and not the best English educated person, you know, not the best local person.
Oh, yeah. Well, I think there is an understanding that you have to be based in Britain.
Of course, yeah.
But I agree that you
bring a big new DNA to this position. Then I mean, you bring.
Yeah, I think it shows a strength and openness of British society,
and the meritocracy,
British science, and that it's a meritocracy. They don't care where you're from.
Yes.
You know, but you know they choose you based on some qualities that they think are important. Yeah, that's true.
Well, some of the people in Cambridge, some of my friends, they commented on you that you are not political. You know, they mentioned you as, you know, as a merit and science person and not a political guy. You know, that was
that I think has worked out in my favor.
Yes.
Because when I have to talk to about Brexit, I have to talk to very academic people who have a certain point of view, but I also have to talk to these very strong Brexiteers, you know, who are part of the government and so on.
Yes.
And so I have had to take a very narrow, objective view when I discuss these things. And so I think that puts me in a position, both sides tend to trust me because they don't see me as having an agenda.
Yes.
Okay. If I have an agenda, I keep it to myself. I don't sort of bring my own personal views into the discussion. So I think people have appreciated that. That's true.
And I mean, you have some time left as president of the Royal Society. So do you, will you focus mainly on the virus situation now, or?
Sorry, for the rest of the time in the Royal Society? Or?
Yes.
Well, there are a number of things. So one is, of course, we will continue to work on issues related to the pandemic for the next month or so, next few weeks. But then after that, I will focus on a few things. One is, I'm very interested in broadening the educational curriculum in Britain.
Oh, yes, science.
You know, so you know, Britain has one of the narrowest educational curriculums in the world. Okay, typically, you know, a high school student only takes 2 or 3 subjects. You know, about 3 subjects at best. The average is actually 2.7. I mean, that is really not fit for the modern world, where you really need a very broad education.
Austria, Germany, we have 10 or 12 subjects until 18.
Absolutely, and that is the correct way to do it. And more importantly, this actually goes all the way into university. You know, at university, what happens is university departments often select you. So they don't really care what else you know. They only want to know if you're in mathematics, do you know enough mathematics to study in our university? So that also is narrow, you know. So I think education needs to be broader and more flexible at all levels, and perhaps less exam oriented, you know. So that is one of the remaining things that I want to try and give it. I've been working on it for a long time, but it got derailed by Brexit and by the pandemic. But in the background, we've been trying to push this agenda forward. And you know, there was this big fiasco with the A-level results and GCSE results in Britain. So perhaps this is a good time to get people to question, how are we educating our people and what should we be doing about it? And then a second question is about diversity. You know, not just in the Royal Society. The Royal Society is, you can think of a fellowship of the Royal Society as sort of a pinnacle.
Yes.
Once you have actually had a great track record and accomplished a lot. But let's face it, most scientists are not going to become fellows of the Royal Society, but it doesn't mean that they're not good scientists. You know, they're often doing very good science. And so in the broad science community, how do we encourage all sorts of people? Now, in the past, a lot of the focus has been on gender equality, getting more women into science, especially into underrepresented areas like engineering, computer science, physics, etc. But also the attrition of women as you go higher up the scale. How do we deal with these? But there's been a lot of emphasis on that, and that's something people are very well aware of. But if you want to know where the situation is really abysmal, it is certain minorities like African origin, people of African or Caribbean descent. They're hugely underrepresented in science, as are some other ethnic minorities, So perhaps, with all this new movement now about Black Lives Matter and the history of the slave trade, etc., there's a cultural shift right now. And maybe this is the time to also look at how we could really try to motivate and nurture talent from all parts of society.
Okay. Maybe the next question I had prepared here was, you in your book you write a lot about your impressions at MRC, and also you comment on the meritocracy of Trinity College, and now you are Royal Society, which is also merit-based. So the question here is, you comment a bit on this in the book, but the question is really, what's a secret sauce which makes such organizations so much more outstanding than others? Like, MRC has like 35 Nobel Prize, and like Trinity College has more Nobel Prize winners than many countries have.
Although I have to say, some of the claims, people only had a 10 years connection. I would say there are about 17. The MRC.
Okay.
Oh, MRC nationally, you're right, has produced more than 30 Nobel laureates, but not the Laboratory of Molecular Biology has produced 17 Nobel laureates, but that's quite a large number for a relatively small institute. It's more than most countries. I think if you were to look at what is the secret, I would say there are a number of things. One is good mentorship. Okay, there's a tradition of doing excellent science. That means that you have people who are established, who are doing really brilliant science themselves. So when they train the next generation, they're training them in both how to think critically about problems, but also how to pick problems, and also when to give up on a problem.
Yes.
If things are not working, it could be for two reasons. One is that it's too difficult or premature. In that case, maybe you should give up. Or it means that actually you're trying the wrong things, and maybe you need to switch directions, or maybe you should just keep persisting and things are working. So this all requires judgment, and it can only be passed on with some sort of tradition of how to do science. And a second is that this may sound ageist, but I think people are most bold and creative when they're relatively young. And one of the things that Britain does is it gives people independence at a relatively young age.
I mean, that's why I went to Britain from Germany, because you can.
Although Germany is changing, Germany has learned that they should do that, and so now if you go to Frankfurt or something, you see young people.
I could be principal investigator in Cambridge like 10 years earlier than I would have been in Germany.
Yes, but that was then. I think now, though, maybe things are a little different. In Germany, they have group leader positions for young people and so on. So I think that's another reason. And a third is that despite all of the, questions about it, I think Britain fundamentally is a meritocracy.
Yes.
And so if you do well, people recognize that and reward it. So
that was always my experience. Yes. Yeah. It's okay. And I mean, the last question maybe is about the virus situation. And I've seen on YouTube, I've seen an interview with Bill Gates who was saying something like, you know, we could spend $1 billion more on building a research base and a platform for vaccines and virus research. And then, you know, this would affect trillions in the economy. And this virus problem could have been it was foreseeable that we would have such a virus problem. I mean, it's not a
yes. I think that's a little unfair. I think Bill Gates is partly right. There's certainly, he predicted, I mean, there's a famous YouTube video from 2015 where he said the biggest threat to humanity is not a nuclear attack or terrorism or that, but it's probably a viral pandemic. Okay, and he also predicted that it would cost the economy a huge amount.
Yes.
And so in many ways, he's completely right. But knowing that, you know, viruses are a big threat doesn't mean that when a new virus comes along, you're going to be instantly able to prepare for, deal with it. Because each new virus will have different properties. It may propagate in different ways.
Yes.
You will need a different vaccine, and that takes time to develop. First of all, when you get a new disease, it's not always obvious that it's caused by a new agent. That takes some time to establish.
Yes.
You know, if people are dying of pneumonia, well, there are hundreds of reasons why people get pneumonia,
sure.
right? Including old bacteria, you know. So to establish that this pneumonia is caused by a new virus,
yes,
and then isolate the virus, and then figure out how it works, how contagious it is, what its routes of infection are. Those are all complicated questions.
Yes, yes.
But what we can do is be much better prepared for it. We can be prepared with reserve capacity, both for diagnostics, for molecular biology, but also reserve medical capacity and reserve public health capacity. And when new viruses emerge, we can then very quickly put together all these tools we have at our disposal to fight it. So I would say it's partly right, but it doesn't mean that if a new virus comes and we're prepared, nothing will happen that will immediately solve it. It will still take time to figure out how to contain it and figure out a cure for it.
But there were a number of similar viruses here in the past, which all seem to have come out of China and affected the countries here. And, you know, the number of people infected and also dying in Japan is much, not just Japan, also in Korea and Taiwan and the countries here in this region is much, much lower than in Europe and UK. And I don't understand the reason. And some people say that there's more immunity here because similar viruses already have attacked here.
That is a theory, and it is a theory that was particularly applied to Vietnam, where for a long time there were no deaths. Okay.
Yes.
But I think that's only part of the part of the reason. I think one of the reasons that countries in East Asia have done well is that they were exposed to the earlier epidemics like SARS and MERS, etc.
Yes.
And so their public health systems were very prepared for a new pandemic or a new epidemic.
That's right.
Yes. And so they very quickly swung into action. They isolated people, quarantined them. They did testing and tracing in Korea. They did impose all sorts of rules about social engagement very, very early on.
Japan has a very strict isolation rule. Japan classifies infectious diseases like 1, 2, 3, 4. And this current virus is classified, I think, number 2. And people who are sorry, 2, I think. And people who are diagnosed diagnosed with this virus are by law isolated in facilities.
Yes. Well, that's true. That's true now in the West as well. One of the problems is compliance. And I wonder if socially and culturally, people are more inclined to comply with regulations like self-isolation, you know, in countries like Japan or Korea and so on. And, you know, Britain and America are both highly libertarian countries, okay? And I think libertarianism is often a good thing. It means a disrespect for authority and, you know, rights for individuals and so on. But in some situations, libertarianism doesn't work very well. And it's when you have like situations like pandemics, then you need to shift to a more communitarian attitude and realize it doesn't mean you're giving up your libertarianism forever. It's just for the duration of this problem. And how to get people to shift their thinking from libertarian to communitarian, that's, you know, beyond my ability.
Yeah, I sent you this email concerning your discussion. I saw your headlines about the masks. So could you get resonance to what the message you were bringing there?
Oh, I got a lot of support,
yes.
But I also got a lot of hate mail from these people who don't like to be told what to do and threatening me and so on. And so I think, you know, people have to understand masks are not a political issue. They're also not a panacea. Just because you wear masks doesn't mean the epidemic is going to suddenly disappear. Because we know, for example, Beijing had a second lockdown. Everybody in China wears masks, you know, and they're required
to.
Yeah,
everybody. I mean, not in this room now, because we kind of trust each other. But as soon as I leave this room, I put on my mask immediately.
Yes. But nevertheless, Beijing had a second lockdown.
Yes.
In Hong Kong and Singapore, there's a resurgence of cases.
Japan also. Japan has a second wave now, but...
Exactly. Even though people are wearing masks. And what that shows is that masks can be helpful, but they cannot solve the problem by themselves.
No, they just reduce the...
They just reduce the transmission rate, you know, somewhat. And so it's a good... you know, I think, so it's not a political issue. So I don't understand some of the attitude of the people who, especially in the US, you know, if you're a conservative Republican, you don't wear a mask. If you're a liberal Democrat, you wear a mask. That's silly, you know, because it has nothing to do with politics.
I mean, Japan is a political country. There's a lot of politics here. Actually, today, I think Prime Minister Abe is going to resign just 200 meters from where we are right now. His Prime Minister's office is just right next. But in Japan, I've never heard any political discussion associated with masks. Never. I think nobody has even thought about this possibility to make them political. If you like, maybe we have a few minutes left. So if you have a little bit more time, maybe we can have some discussion here with...
Sure, absolutely.
If you like.
No, that sounds good. And you can sort of move the camera to whoever's asking the question.
Okay.
Dr. Ramakrishnan, thanks for your time. It's really interesting. I'm Peter Kilner, as Gerhard said. I'm a lawyer, retired lawyer, but I once did natural sciences, although I've forgotten most of my inorganic chemistry, I'm afraid.
So unfortunately have I.
Do you describe you and your family as failed physicists? Is that right?
Yes, that's right, yeah.
Yes. I think one of the other goals that you had, I read when you started as president of the Royal Society, was to try and widen science's, people's knowledge of science in society generally. I know you've been sort of waylaid by Brexit and COVID, but do you think that there's, to any extent, all the discussions around COVID have promoted science in society?
I think so. I think people are now very curious about what is this coronavirus, what are viruses, how do viruses work, what is immunity. So a lot of scientific questions related to the pandemic. And lots of people are going on to television and other news media and on YouTube and talking about the science behind disease and viruses. So I do believe that it is enhancing public interest in science. Of course, you know, I was trying to do that all along. For example, I wrote a popular book about the ribosome, and I've given countless talks to the general public and lots of book festivals about, you know, molecular biology and the ribosome. And the Royal Society has a very active program for public engagement. And we have now 2 annual lectures, which are given for public engagement in science. So I think it's very, very important, because we're going to live in an increasingly technical, science-based or knowledge-based society. And so it's very important that the public should have an understanding of how decisions are made on matters that affect them and what's the basis for them. And they need to know that. So I'm a big fan of public engagement, and also just sharing the excitement of science with the public, and especially with young people, many of whom might go on to become scientists.
Thank you.
Yes. I have just common question as a non-expert about a riposome.
Yes.
You have determined a riposome by X-ray and by crystal.
Yes.
But in the human body,
Yeah.
The ribosome is very different shape. Why?
No, it's actually not in a different shape. That's we know that now because when the structure was determined, you have to realize that these biological crystals are almost 50% water. Okay. In fact, my ribosome crystals were about 70% water. So they're very loosely assembled into a crystal lattice. So the shape doesn't change much. You know, they're crystallized under very gentle conditions. It's not like a physicist, you know, who melts something and then, you know, lets it freeze again and to form crystals. This is a very gentle way of making molecules come together to form crystals. And the reason we know that it doesn't change the structure is we now have the exact same structure using electron microscopy, where the ribosomes were in solution and just rapidly cryo-cooled, and then the structure investigated, and the structures are almost identical. So, yeah, so I think this is a very old, even from when Max Perut solved the first protein structure, this is a question he would be asked. How do you know that this structure represents what it is in solution? The other reason we know that is from the structure, you can predict how it would work. And then you can check those predictions by biochemical experiments. And generally, the structure is a very good guide to how things work. So that's no longer an issue in structural biology.
This is good. Yeah.
If you have okay, hello, Professor. I'm Hazarika. I'm from engineering actually, some civil engineering background. I was in Trinity College last year for about 10-12 days. Basically, my research is soil mechanics. Probably haven't heard of it. It's very, very traditional engineering. My question is very general.
Yes.
Like now, due to COVID, many students are, I think, they are getting less motivated to study and research. And our university, Kyushu University, in a survey, 40% of the students are now suffering from loneliness because of this situation. So, do you have any suggestion to motivate the students not to what to do for the future?
Yeah, I think it's a very difficult problem. You know, especially young children, it turns out, are suffering a huge amount because humans, especially young humans, are very social animals. Okay, we need that interaction. And even those of us who are older, we're not usually hermits, you know, we like to interact with other people. So I'm not a psychologist, so I don't know what the solution is. I think we need to figure out ways to do two things. One is to allow people to interact as much as possible, and preferably not just online, but actually in person, even if they're wearing masks, but you know, if they're outdoors and very well ventilated and staying a little bit far from each other, they can actually see each other without masks and so on. So I think there are lots of ways to encourage interaction in safe ways, and even online interaction, you know, making it personal and, you know, talking about their lives and so on. The other is to assure them that this is a very temporary phase, that, you know, scientists all over the world are working very, very hard on developing long-term solutions like vaccines and medicines, and reminding them that previous pandemics typically lasted a year or two, and then life returned to normal. So they won't feel, I think part of the problem is they don't feel that there's any end to it, that, you know, they're sort of stuck in this weird limbo forever. And that's a very discouraging and depressing feeling. So I think people should realize that eventually things will come back to some sort of normality.
Okay, thank you very much. Thank you.
If you don't mind me going back to ribosomes again, I was fascinated by you were talking essentially about the factories that make the factories. And I had a couple of questions that are sort of related. So if I ask you them all, you might be able to answer them all in one go. I'm interested in how low level an organism would have these ribosomes. And we were talking about single-celled organisms like amoeba. Would they have them?
Every single cell needs ribosomes. Every single life form needs ribosomes, because there is no way to make all the thousands of proteins that actually carry out life's functions and even give organisms their structure without being able to take genetic information and use it to make proteins. And that's been made by the ribosome. That's why the ribosome, it's one of the oldest molecules in the cell. It probably predates the modern version of the cell, predates certainly life as we know it. And even viruses, which some people think of them, I think of them as alive because they're genetic information that replicates, and you know, everybody who gets infected realizes viruses, like bacteria, divide and, you know, cause disease and so on. So I think of viruses as alive, but some people argue that they're sort of on the borderline. Well, viruses don't have ribosomes, but they can't survive except by using the ribosomes from the host cells that they infect. So even viruses could not multiply without ribosomes. So that's how ubiquitous they are.
So can ribosomes have an effect in vaccines or in treatments for this coronavirus?
Not as such. There's an interesting thing about coronaviruses. They make a protein. One of the first proteins encoded in the coronavirus genome is a small protein that goes and blocks the human ribosomes from making other human proteins. And they now understand how it blocks ribosomes.
This is what the antibiotics do for the...
yes, sort of. But what we still don't know is how then do those ribosomes how does a virus allow the ribosomes to make its own proteins? You know, you can understand how it blocks it from making other proteins, but then why doesn't it stop its own proteins? That's still an unanswered question.
The other questions that are kind of related to my, you know, how low does the organism go? When an amoeba splits, then I assume that these ribosomes must be fully created before the amoeba splits, doesn't it?
Yes, what happened? I'll tell you what happened. So before splitting of a cell, the cell usually expands, you know, so it's doubling everything. It's doubling its DNA. Okay, so it's made 2 copies of its genome where there used to be only 1. And then the chromosomes split apart and then the cells divide. But when the cell grows in size, it's also making more ribosomes. And so it's before the split that the cell is making lots and lots of new ribosomes. And then the cell splits, and each half gets roughly its share of ribosomes.
How long does it take for a ribosome to be created?
I don't know what so that's another question. I should note down all the things that I get asked that I keep forgetting to look up. So I don't know how long it takes to assemble a typical ribosome. I'm sure it varies a lot between very fast growing organisms like bacteria and much slower growing organisms like human cells. But it's an interesting question. I need to look that up. My guess is it would be of the order of minutes.
So, I mean, in principle, then you could, I mean, if you're talking about minutes, you could possibly then see, you know, if you had the right methods, you could see the ribosomes being created piece by piece.
Yes, that's right. That's right. So that's the beauty of cryo-electron microscopy. And what they can do now is they can take growing cells and they can get a sample of ribosomes that are all partially assembled. Now, in that class, there'll be assembled ribosomes that have reached different states. So some of them will be at an early stage of assembly, some of them will be further along, until you get the final mature ribosome. And so if you're, you know, good about it, if the samples are good and your computation is powerful, you can sort them out and then look at the structures along different stages of assembly. This would be a little bit like taking snapshots of a factory that's being constructed. You know, the earliest one would be just laying the foundation stone or even just digging a big hole, right? And then slowly the walls go up and the, you know, roofs go up and the, you know, internal walls and then the wiring and plumbing and so on. So I think that's a way of getting these snapshots, and that's a very active area, as I mentioned before.
There's no way to actually then see the ribosomes being created rather than a snapshot of just seeing like the little pictures.
Well, it's you know, then you would have to follow an individual molecule at high resolution. And the problem is you can look at an individual molecule, but there's not enough. What would you see it with? You need something has a resolution that allows you to see it in detail. Well, that resolution is electron microscopy. But if you were to fire your electrons on just, first of all, when you look at an electron microscope, the sample is frozen. It's not actually going to be able to do anything. If it's not frozen, the amount of damage you would do in just the first snapshot would just destroy the molecule. So there isn't a good way to monitor it. But there are other ways to monitor it. For example, people can attach fluorescent reporters to different parts of the molecule, and they can see during the assembly, this molecule must condense. And so the 2 reporters might come together. So they can do that sort of thing. But that's only after you know roughly what the stages are, because otherwise you wouldn't even know where to put those reporters, or you wouldn't know how to interpret it.
Thank you so much. I think we've used up your time now. Thank you so very much.
This has been fun, and well, glad to have chatted with you. It's really a lot of fun.
Well, we did what you recommended to overcome the isolation of this virus crisis.
Right.
So we are together and we are with you. So thank you so very, very much.
Okay. Well,
thank you so much.
Thank you.
Bye.
Bye-bye.
Bye.